Stealth-adapted viruses as an inadvertent consequence of polio vaccines. Most members of the biomedical research community have learned not to criticize vaccines. Indeed, they sincerely believe in the safety and efficacy of vaccines. Sustaining this view has been a lack of disclosure by government officials of actual long-term hazards of vaccines. An example for which I have some experience was the decision to use freshly harvested kidneys from rhesus monkeys to produce polio vaccines. Cells from the monkey kidneys were cultured and inoculated with the polio vaccine viruses. An early finding was that many of the rhesus monkeys used to provide the kidneys were infected with Simian virus 40, SV40. This particular problem was addressed in the early 1960s by switching from rhesus to African green monkeys. It was subsequently realized that both rhesus and African green monkeys were also commonly infected with their own type of cytomegalovirus. In a 1972 joint study by the U.S. Food and Drug Administration (FDA) and the polio vaccine manufacturer, kidney cell cultures from 11 monkeys were set aside from polio vaccine production and tested for the presence of other viruses. All 11 cultures grew African green monkey simian cytomegalovirus (SCMV). This information was withheld from the public, arguably because there had been no reports of cytomegalovirus infections occurring in polio vaccine recipients. Nor was this information brought to light when I informed the FDA that the virus, which I had repeatedly cultured from a woman with the chronic fatigue syndrome (CFS), had unequivocally originated from SCMV. What made this virus particularly noteworthy was the absence of any inflammation in the patient or in subsequently inoculated cats. The cats became acutely ill with evidence of severe brain damage. Interestingly, the animals showed clinical recovery in spite of the absence of immune-stimulated inflammation. By improving the virus culture methods, it became clear that the vast majority of CFS patients and many other patients with neuropsychiatric illnesses were virally infected. Yet the viruses were not being effectively recognized by the cellular immune system. The infecting viruses were designated as being stealth-adapted, with some clearly originating from SCMV. Children with autism were also shown to be uniformly stealth-adapted virus-infected. Public health officials continued to remain disinterested, especially in response to the added suggestion that the AIDS epidemic possibly resulted from the testing of cytomegalovirus-contaminated experimental polio vaccines in African chimpanzees. Public health authorities did, however, respond to learning about my testing of blood donors in 2002 for stealth-adapted viruses. They needed to affirm their belief in the safety of the nation's blood supply by arguing that my reporting on these viruses had put the nation's health in immediate jeopardy. My further clinical testing for these viruses was legally prohibited. This demand gave ammunition to a disgruntled patient to begin pointless legal action and to the founder of a CFS patient support group to poison the internet with accusations of fraud and patient exploitation. In reality, I have never received personal income from the research. The initial prototypical stealth-adapted virus isolate had been largely sequenced, as had small regions on 4 other viruses. In the more fully characterized virus, there are deletions or mutations of the genes coding for the relatively few cytomegalovirus components that are normally targeted by the cellular immune system. There are also additional genetic sequences of cellular and bacterial origin. These renegade sequences may be necessary for the virus to regain its infectivity. Rhesus monkey cellular sequences have been incorporated into 3 of stealth-adapted viruses for which there are limited sequence data. In one of these viruses, some of the original monkey cellular sequences have been exchanged for human cellular sequences. These findings are providing a new conceptual understanding of viruses. They can essentially become disguised as self or as a bacterial infection by incorporating either cellular or bacterial genetic sequences, respectively. Molecular methods applicable to the identification of conventional viruses are not as reliable as performing sensitive virus cultures. Of major significance is that the viruses can potentially act as carriers of disease-inducing cellular genes between individuals and even between animals and man. Stealth-adapted viruses have helped uncover a potent non-immunological antivirus defense mechanism. The mode of virus suppression has been linked to an alternative cellular energy (ACE) pathway. This non-food source of energy is expressed as an added kinetic quality of the body's fluids. A greater willingness to understand, rather than to dismiss, the concept of stealth adaptation would have enabled public health to respond to the COVID-19 pandemic in a more effective manner. All of my efforts to communicate with the senior officials, including numerous emails with attachments and phone calls with staff members, have so far been unsuccessful. Broader discussions on this topic in social media may be helpful.